Welcome back to Chapter 3, Part 3. In this part, we're going to be learning all about lipids. Okay, so lipids, that's another word for that is fats. It's not all lipids are fats, so we're going to talk about that, but sometimes we just substitute the word fat for lipids because that's probably one of the most important categories that we think about, especially when it comes to your diet. The one thing about lipids is that they don't form true polymers. Remember that polymers or building blocks are monomers, and we don't really have that in lipids. And so we'll talk a little bit about the structural nature of lipids in a minute. One thing that's really important about all lipids, no matter what kind we're talking about, is that they have little or no affinity for water. So we use the word hydrophobic because they are made up mostly of hydrocarbons. If you don't remember what those words mean, hydrophobic and hydrocarbons, you might want to go back and watch some of the previous videos. We talked about hydrophobic quite a bit in Chapter 2. So what we have here is a lot of nonpolar covalent bonds. That means that fats separate from water because water molecules like to hydrogen bond to each other. So they're like busy hydrogen bonding to each other, and that means that the fats are not being hydrogen bonded to that, and so they're separate. They are separate from water. So we say like oil and water don't mix, and that's all because of the hydrophobic nature of lipids and the hydrophilic nature of water. Okay, so there's three biologically important lipids, and those are fats, which I mentioned before. There's also something called phospholipids. Maybe if you're paying attention, you could figure out that phospholipids have a phosphate group in them. And then there are steroids. Let's go ahead and look through each of those and get to know them a little bit better. The first one that we're going to talk about are the fats. Fats are lipids that are made up of two basic smaller types of molecules. Now remember, this isn't a polymer because polymers are made up of repeating monomers. But there are still subunits. So our subunits are the glycerol molecule right here, and then we have something called the fatty acid. So it's a fatty acid because it's got this acid group there. But when the fatty acid combines with one of those carbons on the carbon chain of glycerol, what we get is a dehydration reaction. And we talked about dehydration reactions before. And so what we have here is a fat. Now, the rest of that glycerol molecule will be attached to generally speaking, it will be attached to other fats, and so other fatty acids. And so here we have three fatty acids attached to a single glycerol molecule, and the name of the bond right here is called an ester linkage. So this covalent bond is called an ester linkage. Do you remember what the name of the link is for carbohydrates? If not, you might want to rewind and check it out. Fatty acids do vary in how long they are. So some of them can be shorter, some of them can be longer. They also vary in the number of double bonds that they have. And so you probably have heard the terms saturated fats and unsaturated fats. And so if you have a saturated fat, which we see right here, those have the maximum number of hydrogen atoms possible, and there's 0 double bonds. We say it's saturated with hydrogen. This is an unsaturated fatty acid, and it's unsaturated because it's got this double bond right here. And when you've got a double bond, if you remember what happens to those carbons is it loses some of the hydrogen. There's not as many hydrogens. So it's unsaturated. It has a double bond, or maybe 2 or 3, but it has fewer hydrogens, so unsaturated. Now, if you look at fats and fatty acids, and you think about that term saturated and unsaturated, we know that saturated fats are solid at room temperature, And this is why. So saturated fat's like butter, right? That's solid at room temperature. If you think about why, though, look at this. Look at how straight this line is. This is, I know it looks crooked, but overall, we have a fairly straight line of hydrocarbons. And the next one down is also a fairly straight line. And this one's also a fairly straight line. And so if you look at the space-filling molecule, it really is pretty straight. And that's what allows them to stack on top of each other really closely in butter. And that's why it's solid at room temperature. Most animal fats are saturated fats. On the other hand, plant fats and fish fats are often unsaturated. Now, fish are animals, so this is one of the exceptions to the most animal fats are saturated. Fish fats are not saturated, they're unsaturated. Look at what happens with an unsaturated fat. Okay, this one's saturated and this saturated, but this entire fat right here does have this one double bond. And this double bond gives it a little crookedness to the overall molecule. You can see that right here, it looks pretty crooked, which means that at room temperature, these cannot stack on top of each other really nicely. And that's what makes it liquid at room temperature. It's literally the chemical structure that decides whether it's solid or liquid at room temperature. Unsaturated fats can be treated by heat or other means and can become unsaturated. So here's an example of an unsaturated fat that has been allowed to become, sorry, I said that backwards. The unsaturated fat has been converted chemically. And so what we have here, this is called a cis fat molecule. It's cis because the hydrogens are on the same side. And cis fat molecules are actually pretty good for you. Those are the ones that are found in nature. But sometimes when you're doing the chemical processing, what you get is a trans fat. The trans fat has the hydrogens on opposite sides. And so these trans fats, look how straight that molecule is compared to the cis fat with the bend. The bend is good for you. The straightness is bad for you. So when they're chemically done in laboratories, they get some cis and some trans fats going on there. And these trans fats are really, really bad. They contribute to coronary heart disease. So it's best to avoid things like margarine, which has a lot of trans fat in it. So function of fat, the main function of fat is energy storage. Well, looking at this walrus here that has a lot of fat on its body, this animal needs a lot of energy, protecting it from the cold, breaking down those fat molecules and making some ATP, which we'll learn about later. But yeah, lots and lots of energy being carried around by this gorgeous walrus. The next kind of lipid we're going to talk about is the phospholipid. And phospholipids are extremely important for one thing, and that is they participate in the formation of cell membranes, also known as plasma membranes. All right, so let's take a look here at what a phospholipid is. And remember our glycerol molecule, which we can see right here. Our glycerol molecule, oops, I'm sorry. Let me see if I can just draw on here. Oh yeah, there's our phosphate group, and here is our glycerol molecule right there. So we only have two of our hydrophobic fatty acids coming off of that glycerol backbone. There's only two of them. The third carbon, which we see right here, is attached to a phosphate group, and then there's some other stuff up here which is called choline, but we're focusing right here on the phosphate part of the phospholipid. One phosphate and two fatty acids attached to the glycerol backbone. When phospholipids are added to water, they form into something called a bilayer. Here's the thing. I'm going to go back here. in this phospholipid, this is called the head. This glycerol and the phosphate and the choline, this is the head of the molecule. And these are the tails. These, oops, sorry. Come back. Pen, there you are. And these are the tails. The tails are hydrocarbon based, and so they don't like water, right? And the heads, they've got a lot of ion regions, like here, and so they are attracted to water. So they're hydrophilic. When you put this molecule, this hydrophilic head, and these hydrophobic tails in water, the hydrophilic heads are going to be attracted to the water, and the hydrophobic tails, not so much. Those hydrophobic tails form this boundary, or this double layer right here in the middle. We call that double layer a bilayer. And there's your word bilayer right here. So this is a bilayer. Here's one layer, and here's the other layer, and the hydrophobic tails are attracted to each other. This phospholipid bilayer is so important. It forms the boundary between what's inside a cell and what's outside a cell. And that's extremely important for understanding how cells work. Our last category are the steroids. And the steroids are forms of lipids. Notice again that it's not a monomer. They're made up of, the carbon skeleton is made up of these 4 fused rings, 1, 2, 3, 4, and often have different functional groups or side chains coming off of them. This that you see right here is cholesterol, and cholesterol is an extremely important steroid hormone. We often give cholesterol a bad rep, right? It's got a bad reputation. That's because if you eat too much cholesterol, it can lead to heart disease, and that's bad. Heart disease is specifically arterial sclerosis, which is how you pronounce this word, arterial sclerosis. So 1, 2, 3, 4 rings, and the side chain, if it's floating free in your blood, not so great. But if it's in your membrane, if it's in your cell membrane, that's extremely important. It helps, as we'll learn in later chapters, to maintain the flexibility in various temperatures. So our next lecture part, chapter 3, is going to be part 4, where we're going to learn all about proteins.